PhD supervisor, Dr. Frances Skinner, to our KCNI speaker series. Dr. Skinner is a senior scientist at Krembil Research Institute and a professor at University of Toronto. She obtained her bachelor's degree in mathematics at University of Waterloo, her master's and PhD in biomedical and mechanical engineering at University of Toronto, and did 4 years of postdoctoral work at Brandeis University and University of California. Her work, at least from my perspective in more recent years, has included a broad array of computational modeling, uncovering the mechanisms governing cell excitability in inhibitory hippocampal interneurons, as well as the underlying mechanisms of intrinsic theta rhythm generation in hippocampal neuronal networks. So Dr. Skinner has a saying, which is to neither ignore the details nor be consumed by them, which I think well encapsulates what makes her such a great computational modeler, collaborator, and mentor to trainees. Today, she'll be presenting some of her work on modeling inhibitory interneurons in the hippocampus. So please join me in welcoming Dr. Frances Skinner.
Okay, well, thank you very much. And thank you for the very kind introduction, Dr. McCraig. So today, the title of my talk is A Modeling Story of Two Brain Cells. And I'm very pleased to have been invited to this speaker series, so thank you for that. And as you can see in my first slide, I have a giant logo of the Skinner Lab, and that's the logo is thanks to Jeremy Lefebvre, another KCN hubber. But I started using this just at the previous talk I did because I always say, and this is just a reminder about how any work that I present, of course, is a function of the lab. Of course, there's a specific people who've done the work, but within the lab, all the discussions and the criticisms and feedback and discussion and I don't know if I would say arguments sort of make a difference. And so I also really like the idea of developing bridges because really this is what it's all about. So I want to start with the acknowledgements in case I run out of time. So as I mentioned, all present and past lab members and collaborators are key in any work that comes out of the lab, in my opinion. But for what I'm going to talk about today, specifically Vlad and Alex, it's their work that I will be talking about in terms of these two brain cells, which I'll talk about shortly. And of course, the collaboration, the experimental collaborators are George Lawrence and Lisa Topolnik. Excuse me. And of course, none of the work would be possible without sort of financial and institutional support. So I put all sort of the logos here that made this work possible. So thank you for that. And so as Alex said, it's a balance and neither ignore the details. I don't know if I put that particular slide in, but I wanted to share this slide because in my mind, yes, it is always a balance, but more specifically, it's a balance between the experimental work, the model development with it, the theoretical analyses that we want to harness, and of course the computation and use that we need depending on what sort of your model encompasses. But these arrows that are bidirectional are intentionally that because really we want to sort of bring these all together. And I don't think we're there yet, but I mean, the point is to just try. And so the two quotes that I wanted to sort of share with you before I dive in are these. So this first one is from an opinion in 2015. And there's many interesting things in this particular article. But in one quote that I'd like to take from it is that principled model building will be ever more important in this era of big data, which clearly we're at, as it is only the principled model construction and evaluation that will allow us to understand which details are important for what functions of the brain. And so really, this is this huge challenge that all of us in neuroscience are faced with. And the question is how to kind of go about this principled model building. And so in some sense, this is kind of the focus of that, hopefully thread that will go through my talk in terms of how we sort of try to do this by discussions and interactions. And the second quote is from this perspective that was just from last year. Well, I guess it's almost the end of 2020. So this was in 2019. And this was by Annabelle et al. And I like it for many reasons, but one of the points is sort of this clear intersection that comes about with computational neuroscience and neuroinformatics, which very nicely we have here in Toronto with the KCNI and our KCN hub at Krembil. And one of the things they talk about, which really sort of brings about why we really need to sort of have this interaction and discussions, is that the mechanistic modeling of biological neural networks mimicking specific brains or brain areas is really in its infancy. So lots of work to do, but I think there's lots of excitement because of the interactions and developments that we're seeing here in Toronto, for example. So I'm going to tell you a story. And so the story is about these two brain cells. But before I sort of launch into that, just to sort of give you an overview of sort of my lab. And so, in my lab, the way I think about it is what we're actually trying to do is a collaborative cycling towards convergence and translation. And in this sort of schematic, I'm sort of showing sort of the aspects, the various aspects in the lab. And I want to sort of point out this sort of cycling aspect because it's sort of always ongoing, where we sort of build these models, whether they're biophysical neurons or microcircuits, where the focus is, with the experimental data. And we want to try and come up with hypotheses and predictions, from the model that involved experiment and feedback. And it's an ongoing process. And even though I won't be talking about this work today, we sort of recently, this work has been submitted in bioRxiv about how rhythms are generated. And Alexandra now, Dr. Hatzikilaminyu, took advantage, really sort of brought this translating opportunity to a core, to fruition, I should say, by bringing together sort of two different levels of model. And I mention this because this allows me to sort of bring about, actually, let me just go back here. So there's sort of a earlier minimal model that sort of had simplified Issakevish type point neuron model. So they had no morphology. And then there's this detailed model from the Schultz's lab that we brought together in the context of an intrinsic theta rhythm. So I just mentioned that because that allows me to sort of transition into making the point that, of course, our brains are made up of all these sort of detailed cell types. And in this particular model that the Schultz's lab put together of the C1 microcircuit, there's many different cell types in here. And one of the cell types that are in here is this OLM cell, which is actually the purple one, and then this IS3 interneurospecific cell, which is not in here yet. So I just mentioned that at the outset to sort of show this transition. And so today I'm going to be talking about these two particular cell types, this where we sort of developed over the years, and try to give you a sense of sort of how this sort of cycle has worked. Okay, so, why do we care about these details of the cells? Well, I think we have no choice because all of us here, of course, care about disease. And the abnormalities that we see in diseases such as Alzheimer's are not just, of course, at the whole brain level that you can see by looking, an fMRI, but it's also known that specific cell types, and here's just an illustration of one particular inhibitory cell, PV cell. Oops, sorry. Let me. Okay. Oh, sorry. Sorry about that. Yeah. So this is one particular inhibitory cell type that has particular ion channels that change in an Alzheimer's mouse model. So this is just to sort of make the point that we have to care about all these aspects, but it's a huge challenge. But we know specifically that inhibitory cells or interneurons, their cellular details are critical. So that's fine and good, but look at all this stuff we've got to worry about. So there's the different morphologies, the connectivity, there's different markers, intrinsic properties. We have to worry about all those things, but at the same time, we also know, as schematized in this particular review, that these different inhibitory cell types, and I've circled the ones that I'm going to talk about today, fire in distinct phase-dependent ways in, for example, on an ongoing rhythm here, showing a gamma and a theta, ripples and sharp waves, or in different behaviors when, say, the animal's entering or exiting in a task, there's a firing of specific cell types. So clearly, we really have to worry about this, but exactly how to go about this is not clear. And this is where sort of this interaction that I was, balance that I was talking about in the beginning comes about. And so I just put this up because, yes, we have to worry about it, but as this group showed in 2018, as far as the CA1 hippocampus is concerned with inhibitory cells, we have many different types. So there's 10 groups, 49 fine scale clusters. And back in 2006, when Buzaki's book on Rhythms of the Brain came out, at that time, he was saying, we really cannot think about the brain as inhibitory cells and excitatory cells in this sort of gross sense. Interneurons, the inhibitory cell types, have changed dramatically in the past decade, and that was in 2006. So we cannot think about it as you can't treat it as a homogeneous collection of neurons providing negative feedback, but they have particular intrinsic properties and particular wirings. And as I just showed you in a schematized view, they play particular roles. Disentangling and trying to understand that is really, what we're faced with, and that we cannot ignore if we want to sort of tackle disease, think about therapeutics and all the rest of it from cellular perspectives. So from a modeling perspective, really, in my mind, this all breaks down to being clear about the goal, the rationale, and the limitations in thinking about this sort of balance. Because in that way, we could really harness and take advantage of theoretical insights, the particular model, how it's developed, what it includes, the particular experiments that are possible, what are the limitations in computation, what makes sense if we want to sort of extract out and fully explore parameter space and the like. And so this modeling story of two brain cells that I'm going to tell you about is in a context of a theta rhythm. So I'll get to that in a little bit more, but before I jump there, I want to mention Alex, who I think everybody here, at least the host, knows, and you see his beautiful artwork in the back. This is a review that Alex put together very recently, and I put it here because even though I will be talking about the OLM and the IS3 cells that we've modeled over the years, there's a clear analogy in the cortex. And so, of course, the cortex and hippocampus is not the same, unlike what some people might try to convince me of. But clearly the connectivity and sort of certain characteristics have overlap. And so in this review that Alex put together, he sort of talks about these common principles from the VIP cowretin, which is these IS3 cells that sort of have a dissonatory role, but we'll get to details shortly. Okay, so theta rhythms are something that, the most robust rhythm in the hippocampus. And today we know that clearly it's not just an epiphenomenon. I think it's fair to say there's a Nobel Prize that was given a few years ago about this theta rhythm really sort of being a functional unit in terms of phase coding. So we've known, and it's been shown in many ways, that you have this phase precession, the spatial coding. So as the animal moves through space, you have this firing of these space cells that precess relative to the theta rhythm. And in this now, I guess, 5-year-old review by Wilson et al., they really sort of bring this to bear, showing that there's sort of this phase coding that occurs with the theta rhythm context. And then way back in 2002, Hasselmo et al. Proposed this function for theta rhythms in terms of encoding and retrieval characteristics. And we know today that this is not just a hypothesis or proposal, it actually seems to be happening thanks to optogenetic and techniques and sophisticated experiments that can be done now. Siegel and Wilson showed that you actually have an enhancement of encoding and retrieval in a phase-specific manner. So this proposal by Hasselmo back in 2002, where shows that in the encoding phase, you have this strengthening from the entorhinal cortex, and in the retrieval, within the CA1, and then there's a weak entorhinal cortex input. This really seems to be happening in terms of the trough and the peak when you think of the theta rhythm as measured in the EEG at the fissure is going on. So a lot of details in there, but really the take-home message is this theta rhythm is this robust, functionally important rhythm that we're going to use as a context when we talk about these two particular cell types. So it's not just a matter of like saying, okay, we have these particular cells and let's just build models of them. It's sort of building them and trying to understand how they contribute to behavior, essentially. And of course, even though that is all in rodents, when they talk about grid cells, place cells, we have these theta rhythms clearly in humans as well. And in this recent article, this is showing sort of theta rhythms in 3 different agents here. So more details there, but I want to now get into the story. So the first cell that I'm going to talk about is this Orion's lacunomosan moleculari, or OLM, interneuron. So these are one of the inhibitory cells in the hippocampus. And this beautiful image from McAfee and Lakai in 2003 shows here's the cell body with its dendrites. And here's this huge axonal arborization that goes sort of to the distal regions of the green, which is the pyramidal cell excitatory cell outputs. So this is a beautiful cell that sort of has been studied because it has a very distinct morphology. And in addition, it's long been known that they have this very strong SAG. And it was in 1996 that McAfee showed this. This SAG, which you see when you hyperpolarize this current, is due to H channels or these hyperpolarization activated inward currents. And so that's been known for a long, long time, the particular biophysical characteristics, but how it's distributed, the density, all these details associated with it is, sort of still a little bit open. And so I'll get into that in a second. So that's great. We have this nice morphological cell. But even way back in 1995, it was proposed by Blasco, Ibanez, and Freund that this cell type, so here is it, this at that time, they're calling it a somatostatin positive cell, synapsing onto distal regions, is playing this sort of switch role because it's synapsing in the same region that the incoming perforant path input. So you have a intrahippocampal pathway as well as sort of sensory input coming in. So it seems to have the ability to play sort of this switch, and that was proposed back in 1995. Jump to present, and we know that this is really happening. So again, tools to be able to identify, in the behaving animal and manipulate and all the rest of it, that Richardson, Leo, and colleagues were able to show directly that OLM cells are important, really sort of gate this information flow in CA1 by facilitating that transmission from the CA3 and the extra inputs from the entorhinal cortex, as referring to. So this cell is playing this role. But this cell has this morphology and has all these biophysical characteristics, and how is this contributing? How is it doing this? And so we set out many years ago to start building models of these OLM cells. And here I'm just showing why we actually built multi-compartment cells. Well, because it was shown by Martina et al. That this OLM cell has very active dendrites. So this is just showing that it has sodium and potassium channels in its dendrites. They're not sort of passive. And so we can't really sort of justify starting off with a point, no spatial representation for this neuron. So we built so the initial OLM cell we built sort of took that into consideration. So we had a multi-compartment model. And I'm not going to talk about details, this was a while ago, but this model existed in a certain capacity for morphology and certain biophysical characteristics. But what we know present day, now this is 2011, as was pioneered in Yves Marda's lab, is there's a huge amount of biological variability. So even though you're talking about a particular identified cell type, say the OLM cell or any other cell that you take, you measure it one day, you have a certain conductance for, value, a maximal conductance value for sodium channels. You measure it another day, it's different. But this cell in the circuit is, of course, doing its function appropriately. So in the stomatogastric ganglion, it's generating this 1 Hz rhythm to move the stomach and to move the teeth in the stomach of the crab. And it doesn't matter what, whether it's 10 or 15, sodium conductance is able to do it. So there's this balance that the cells are able to maintain to do its function. And so this paradigm shift, as Yves called it back in 2011, said, rather than sort of trying to build a single model, I mean, we can't really justify this given that you can have a 6-fold difference in the conductance values, but rather a family of models to sort of try to understand and reflect this variance in the data. And so when Vlad joined my lab many years ago, we sort of said, okay, we cannot just stick with a single model that, single OLM model that we had developed way back to sort of look at these active conductances and dendrite, but really need to do a model database. And so we started along that road, but we did it with a particular question in mind. In other words, when we built this model database, we didn't just sort of build thousands of models. We sort of said, well, what is it we are trying to do? Well, the big thing in terms of the OLM cell models back in 2014, I guess, when Vlad started this work, is we've long known that we have hyper IH channels, hyperpolarization activated mixed cation currents generated by H channels. But we don't know where it is exactly. Is it in the dendrite, soma, distributed certain way? I mean, in pyramidal cells, you have sort of a distribution of IE and IH along the dendritic arbor that sort of ramps up or ramps down, but we don't know for these OLM cells. So he said, okay, well, we have this model. Let's build a model database where we have a reference model, compartment model that we developed previously, and now build it in such a way that we match experimental data when you have H channels in the dendrites or not. And so basically Vlad did that. So that was that specific question we had in mind. And long story short is we had these different models that were representing OLM cells, but because of that, we're able to leverage those databases to actually make a pretty fundamental observation from the model that there was actually a preferred spiking recruitment at high frequency or low frequency theta, which we know exists, is functionally important, depending on whether you had H channels in the dendrites or not. So we were only able to do that because we had these two databases that Vlad had developed. And so this is kind of the strategy that I sort of, and these ideas of how the sort of principal model building and what you're trying to explore and caveats and all the rest of it that I hope will kind of go through this sort of my talk today. And so since we don't actually know, or it wasn't known, I would say, at this point about the presence of H channels in the dendrites or how they're distributed, this sort of says that depending on the distribution, it matters in terms of the actual spike resonant frequency that it could contribute to the OLM cells. And what I would also want to point out by showing this picture here is the way Vlad did this was by building a simple in vivo-like situation. So sort of a virtual network. And so this idea built off, of course, previous work, a figure from Dessesh et al. In 2003, where we've known, so their work and others since then, that if you're in an in vitro situation, so you're just recording in the dish versus in the behaving animal in the in vivo, like the behavior of it's sitting in a different environment. You have sort of a higher level, there's a lot more noise going on in the behaving animal, so the cells are going to respond differently. And so in order mathematically or in a model context, you can sort of create these in vivo-like situations. This is really important because not only does it allow, if you have a multi-compartment model, you could really sort of explore this, and you just cannot do this easily experimentally, but it's very different from if you actually just stick your electrode and create these artificial situations by the soma, because the input is coming in, all in different places in the dendrite. So Vlad essentially did this when he did this work, but sort of did it in a sort of, what I would say, a simplified way, sort of essentially added noise in different parts of the dendrite. But he had this very cool observation prediction. And this was coming out of this cycling and leveraging. So when he did this work, we are using these sort of originally developed OLM cell models. We didn't know if H channels were in the dendrites or not, but he sort of showed that if one population versus the other, we'd have a different high or low spike frequency resonance, theta frequency resonance. So thanks to a collaboration with Josh Lawrence's lab, we said, okay, we know that it sort of matters functionally, but we don't know what's going on. We know we've used these previous models. We also know from other modeling work, which I'm not showing you here, that you can kind of adjust the total conductance, you can adjust things in the passive properties and kind of get different distributions and things to work. So what you really need to know from the experimental data, if possible, is to have the same cell recording, characterize the H channels in the same cell, and build a model of that same cell to really see if you can get some sort of consistency. And so that's exactly what was done as part of Vlad's PhD. And this is the final many titles we went through of this work. And so it's integration of within cell experimental data with the multi-compartment model, and basically predicting that we do have H channels in the dendrites. And so, as I said, I'm just showing you the snapshot, but basically it was a huge amount of work because you're characterizing the H channel characteristics in the same cell from which you're getting the morphology and the passive properties and everything. And so, and you're building models of that same cell and fitting, the data you have. But now you have the constraint of the total conductance of the H channel in that particular cell. And so it was impossible, or I should say it was, the matching was definitely with H channels in the dendrites and not within the soma only. So basically we sort of have the answer. And this is not to say that, we don't need experimental work. There is a really preliminary experimental work that shows from immuno studies that the H channels in the dendrites. But what I would emphasize is even with that sort of experimental work, which you of course want to have, it doesn't tell you about densities and distributions, all the rest of it that you need to sort of build a full model to sort of understand how these cells contribute in a circuit in the behaving animal. So we kind of, this is sort of this balance and bringing everything together that I'm sort of trying to sort of emphasize here. So meanwhile, we have interneuron specific or IS3 cells. So these are another inhibitory cell type in the hippocampus, and that's circled here as taken from this review by Chamberlain and Topoltek. And these inhibitory cell types target only other inhibitory cells. And so here's just a sort of image from Lisa's lab showing specifically that these VIP positive, specifically these IS3 cells, are targeting OLM cells. So that's why I'm bringing the story of the two cells together, and you'll see sort of why different aspects are important. Hopefully, I'm bringing this across clearly. So these interneuron-specific 3 cells seem to be sort of, have this kind of very particular role, but, how and what is it doing, and why, what is important? And so we built models. So this is basically Alex's work I'm going to be talking about. So if you have any detailed questions, Alex is here to answer them, but I will try. So basically, getting into sort of the idea of sort of the biophysical detailed properties in individual cell types, which is really hard to sort of get a handle on. And actually what might be, what I wanted to mention at some point, and so now is a good time, is for those of you who saw Sean's, Professor Hill's excellent talk last week in physiology, where he's talking about all these different cortical cell models. And I mean, there's beautiful work, but the challenge and the details associated with building sort of an individual cell with all its properties is there. And even in those sort of detailed models, when they build it, they're sort of set and they're fitting and they're optimizing and all this cool stuff is happening, but you're setting the distribution. So you kind of need to know what this information is. So here, what we're doing is we're focusing on this particular cell and hopefully will contribute to sort of that sort of big picture, but you kind of need that ahead of time. So part of it is just trying to sort of use the models to get this information in sort of in this cycling fashion that I sort of showed you in the beginning. So here's this IS3 cell, and Alex sort of developed this sort of what he called a semi-automated strategy. I think he had a fancier name before that to come up with the IS3 cell model, multi-compartment model. And again, the multi-compartment, so there's biophysical ion channels in the soma and the dendrites and all the rest of it. And so we didn't have any of that information here for the IS3 cell. And so we sort of built on what made sense in fitting that, Alex really fit to the sort of electrophysiology recordings and, taking advantage of other interneuron specific ideas. And basically Alex's distribution sort of predicted that you had these fast delayed rectifiers. So I'm just showing you, a snapshot in the dendrites. And so Lisa's lab, they actually went ahead and did these experiments, and they actually found that KV 3.1, which is actually the subunit in the fast delayed rectifier, is actually present in the soma and proximal dendrites of these IS3 cells. So bear in mind, this is not a small amount of work. So to have an experimentalist to sort of undertake to sort of explore something particular, to have a particular hypothesis from the modeling work to drive the experiment is really important. And this whole work with this whole collaboration with Lisa's lab has been fantastic in terms of really the interdigitation, which you'll see more of shortly. So there was that prediction that sort of held fruit. And so now, wanting to sort of take and predict and understand how this IS3 cell is contributing in this theta context, we built these virtual networks. So as I referred to in Vlad's work, when he had the OLM cell, he sort of did a simple sort of synaptic noise representation of situating his OLM cell in vivo-like situation. Alex did the same for his IS3 cell, except it was actually a sort of detailed representation. So I circled here because essentially what he did was he fit exciter inhibitor inputs in the dendritic tree as optimized to recordings that we had from Lisa's lab. And this is just showing example output firing of what the IS3 cell is doing and exciter inhibitory current or conductance balances to have it in an in vivo-like state. So this was a massive computational undertaking that Alex did. But what was fantastic is that once we had it, we were able to sort of do a virtual network kind of representation of what these IS3 cells, how are they contributing in the behaving animal. And so in this, I love this image that Alex and I think, well, he had help put together in one of his publications, is sort of really sort of showing sort of the steps, right? I mean, we want to know what's happening in the behaving animal. It's very of particular cell types and how they contribute. And here we're talking about the IS3 cells in a theta rhythm context. But it's really, really hard to have identified cells and know and control when the animal is, moving and doing stuff. So if we have a model and we simulate this behavioral activity by putting sort of this virtual network, we can sort of come up with hypothesis, ideas, and predictions of what's going on. So that's exactly what Alex did. And so in the IS3 cell in particular, what you're seeing here is a polar plot of what the IS3 cell is doing, baseline versus when you have a theta activity going on. So this is a virtual network, meaning you're sort of imposing this sort of thing. It's sort of, first of all, in an in vivo-like state, as I sort of showed you here. Oops. Okay. Yes, here. So it's in this state, and now we're imposing this theta rhythm, okay, in ways where we know the inputs coming in from different cell types. And basically, what the essential prediction from this work is this computational exploration that Alex did was there was a preferred firing at the rising peak. You see this increased firing of the IS3 cell during this theta rhythm. So you have the peaks and the troughs, as I showed you before, and we do care about peaks and troughs. And as I sort of tried to sort of show you before with the Haskell work. And so he had this very clear prediction. And what was, of course, really nice is that in Lisa's lab, now I'm not going to show the details of the experimental work, but at the time, they actually were carrying out these very challenging in vivo experiments, trying to target and understand exactly what the IS3 cells were doing at the time. So, of course, limited data, but this prediction that Alex made exactly held fruit. So it was very exciting. And I think it sort of speaks to sort of this strategy, if you like, of creating these virtual networks to sort of try to understand what particular cell types that are a minority but seem to be playing particular roles are actually doing. And so this was, this work is now published, and it really sort of speaks to this integration and why I think this sort of tight integration and interaction between the labs is really needed. So I say that, and now I'm going to go back to earlier work where we've now taken further advantage. So this is an earlier paper from Lisa's lab, and now this is just showing that the IS3 cells target different inhibitory cell types, the OLM cells that I mentioned already, bi-stratified cells and other ones, but it's mainly the OLM cells that they connect onto. So now, as you can guess, we're going to try to bring our models together and understand something more. So in this work, what they showed using optogenetics was that first of all, the IS3 cells target OLM cells preferentially, and they actually control the spike timing at 5 and 10 hertz, so in that theta frequency range, not 1 and 20 hertz. And what they suggested in this paper was that post-inhibitory rebound mechanisms are what playing the role, because there's an inhibitory input onto an inhibitory cell, and it's sort of controlling the timing at a certain frequency. So they suggest like post-inhibitory rebound in terms of the mechanism that's underlying that. Okay, so we have this OLM cell now, these sort of very, what I like to call state-of-the-art models for the OLM cells, even though the reviewers don't like that, but that's another story. But really, these are cells, those different OLM models that I talked about were characterized, passive properties, morphology, and eye characteristics from the same cell. And we had 3 of them, as I mentioned, that were fully realized, even though about 14 recordings, but you need the full set of everything. And so we had full spiking models for 2 of them that Alex sort of took the next stage to bring them to the full spiking models with all the different sodium channels, potassium channels, calcium channels, all the rest of it that we knew. And in this work, which I'm not going to talk about in detail, but just to sort of show, because of having those cells, we can now start comparing in vitro and in vivo like character. Oh, I see something here. There's a question. Do you want to ask me the question, Itai? I saw something come on my screen, but I can't see anything else.
It's okay, Dr. Skinner. We can go over the questions at the end as well. Okay, all right.
Right. Okay, so we had these OLM cell models, these, state of the art that I'm going to call them models because of the specificity and the variability that was captured, but they're all OLM cells. Now, I'm not showing you here, but basically Alex went through the same sort of computational study where he sort of created this in vivo-like state. So here you're just seeing what it looks like, the firing at the top here in the soma, and then this is 4 different regions in the dendrite. And you see, of course, the firing changes. What you're seeing below here, which I'm not going to talk about in detail, is this visualization called current scape that allows you to see how the different biophysical currents are contributing to the firing there. So these are 2 OLM cells, cell 1 and cell 2, and it's important to look at more than one because even though they're both OLM cells, they have different densities associated with them. Okay, so here's IS3 to OLM cell. And what we did now is we took these models, or I should say Alex took these models, and we did a phase response curve analysis. Now, phase response curve, for those who don't know, is essentially you have this oscillating system, and you're giving a stimulus at different phases within that single cycle. So this cycle I'm talking about is the spiking of the OLM cell. And basically, the input in this case to the OLM cell is IS3 input at different phases. The input is also as if the IS3 cell were right of the numbers that we know exist from work in Lisa's lab. But because this is a model, we could also simultaneously access the biophysical characteristics. And so this is what Alex is showing here. So let me just sort of walk you through this a little bit. These top 4 plots are phase response curves. And so the x-axis is actually the phase. So if you think 0 to 100%, so 0 to 360 degrees, however you want to do it, of where the stimulus is given to the cycle of the spike. Now, depending the phase at which it's given, the subsequent spike or the subsequent oscillation is either advanced or delayed. And in the way this is set up, positive values mean it's actually a delay and negative is an advance. So that means what you see here, most of these are actually positive. That means most of the times you have a delay of the next spike. So that means this incoming inhibitory input from the IS3 to the OLM cell is actually delaying the next spike. Now, what's important is the different frequencies that are seen here. So this is sort of just at Brio base so that the OLM cell is at, or the OLM cell is set to be firing at 4 Hertz, 7.25, 15 Hertz. And you see, it matters, of course, in terms of sort of the cycling. But below here is essentially an analysis of all the ion channels that exist in the OLM cell model. Now, I'm not going to walk you through all this, but just to point you down here, which is this color, which is actually the IH, H channel, if you notice, has the largest values compared to any of the currents. So that allows us to sort of say that any phase advance or post-inhibitory rebound that would exist has to be mediated by, is primarily mediated by the lower H channels and only at lower spike rates. Because here, the lower spike rate, here you have a little tiny advance, here you have a bit of advance, and this is only in cell 1 also. So this now gets into the different characteristics. But suffice it to say, if there's going to be a phase advance, meaning that you have post-inhibitory rebound, so that means the inhibition is causing the next spike to happen sooner, it is going to be primarily mediated by H channels, not by any of the other currents. So there's details in there, but I think what I wanted to bring about is the strategy that we were able to do to sort of understand what's going on. Jumping to sort of the in vivo-like state that Alex created in these OLM cells that now allowed us to sort of essentially confirm the earlier work that Vlad did where he sort of made this prediction. So now from that work, we know that the H channels are in the dendrites. So these models have H channels in the dendrites, and that would be at a higher frequency. So there's details in here associated with the different synaptic noise or in vivo light activity, which I could get into, but let me just leave it there for now because I see the time. But essentially, the OLM cells have a spike resonant frequency at theta frequencies when you have the incoming inhibitory input from putative IS3 cells. Not for excitatory cells, not for excitatory input or combination, only for inhibitory input in the in vivo-like state. So going back to my favorite image from Alex, so we have this IS3 cell onto OLM cells when you have this sort of in vivo understanding, what are mechanisms with the biophysical characteristics. And as I mentioned, Alex sort of created this sort of, or simulated this virtual network to simulate behavioral activity. And so now we can go back to what we understood from those experiments that went on in Lisa's lab, about what the IS3 cells were doing in the behaving animal. So as you recall, there was prediction of where IS3 cells fired during the ongoing theta rhythm. What was also seen in that experimental data, in vivo data, is the IS3 cells activated with a delay relative to these theta run epochs. So now Alex took further advantage of his IS3 and OLM cell model mechanism ideas and created this sort of ramp up while the animal is during this ongoing theta rhythm. So again, there's details in here which I'm not going to sort of belabor because I see the time in front of me, and to say that this exploration that Alex undertook allowed him, even though it was in a virtual network situation, to sort of come up with sort of a prediction of what's going on. So, and here it is. So the prediction essentially is that the IS3 cells are actually suppressing the OLM cell spiking, not causing it to spike by post-inhibitory rebound. And that's coming off from sort of that early work and details, which I haven't sort of described to you here. It is also the case that what Alex is predicting from his study is that you needed to have disinhibited pyramidal cell inputs. Okay, so here's what's going on. Here's the proposal of what's going on. The IS3 cells synapsing onto OLM cells, as we know, it's actually suppressing out of phase, potentially putatively suppressing out of phase OLM cell spiking, which because of that, you have a ungating of the pyramidal cell distal dendrites, and it creates this window which now feeds back and can sharpen the OLM cell spiking with recurrent excitation. So you essentially have this kind of, what IS3 cell is actually doing is sort of timing and keeping the OLM cell to sort of spike in a sort of narrow, more precise range. And so the proposed functional role here is that what these IS3 cells are specialized in doing is creating very temporal precise coordination of these modulation pathways. And so with that, let me just sort of recap that, you know, even though some of you may or may not care about this hippocampus as much as some of us, as myself and other people do, but really I think the strategy and the thinking that went into sort of this work here is what is key in bringing it about, because at the end of the day, it's really about the goal. What is it we're trying to do? So we're trying to understand the details associated with these different cellular inhibitory cells and how they contribute to function, and we sort of put the theta rhythm context and all the rest I talked about. So the rationale when one is developing these models has to be clear, but as well as the limitations. So we're using this virtual networks. Of course, you don't have feedback and all the rest of it, but if you wanted to build a whole model with all the detail, as you know, as has been done, you know, as is, you know, undergoing with the cortical network, but again, it's a cycling fashion. Even in that detailed fashion, you have to worry about these ion channel, the biophysical details and the distribution. And so you can't sort of do the kind of exploration you're doing at the same time. So the hope is that this kind of work could feed into sort of details model, as well as maybe simplify and pull out sort of, you know, essential mechanisms that underlie theta rhythm generation frequency from the perspective of these particular cell types. So again, my favorite picture with the double arrows and the acknowledgements that I started with, and because I put in the beginning, I want to sort of end and give people time for questions, and any discussion is welcome. So thank you very much for your attention.
Big applause.
Thank you.
I'll start off with some questions from Itai. He said, "Hi, Francis, do we know anything about how monkey and human hippocampal neurons compare to rodent neurons?"
So, I mean, yeah, so, okay, depends on what, so yes, we know stuff. And so that one figure that I showed you, so Joshua Jacobs has done a lot of work with the human theta and looking at, so first of all, in humans compared to rodents, and actually I assume that's true for monkey to a certain extent, the theta frequency is actually lower. But there was a paper that actually just came out that showed you still have this sort of higher low theta that is sort of functionally relevant in terms of participating in certain behaviors or not. So that was Cote et al. So since I can't actually interact with you, Itai, to sort of see if I'm kind of asking you the question, oh, there I see, intrinsic properties dimension. So yeah, human is their larger cells and all the rest of it. So you know all about that in terms of the cortex. So I have not sort of specifically sort of delved into modeling human inhibitory cells in the hippocampus, but yeah, I mean, there would be the sort of similar differences. So, I mean, that's why, again, I think sort of the context and how they contribute. So I mean, I think it's reasonable to assume that, you have these different cell types in humans and rodents and monkeys. So in terms of how they would contribute, sort of having the switching mechanism, there's going to be a similarity there. So the details with the intrinsic properties and dimensions will differ, are definitely different without a doubt. But I couldn't tell you something particular. But I guess the thinking and the strategies here is because we have access to the rodent, as you know, you can get sort of these fine details. And so if we get a mechanistic understanding of these balances and what biophysical property is actually contributing, then it sort of directs you what to kind of potentially look for in the human situation, where it's much more limiting of what you can do and what you can look for. And I think the work that we did with Lisa's lab sort of really points to that this is a very fruitful way to go about. And again, like you could look at, so this whole transcriptomics is very exciting, right? I mean, I know Lisa's lab, and maybe in collaboration with you guys there too at the KCNI, looking at these details. But again, the point is, what to look for. So it's developing these hypotheses in that way. So does that kind of answer your question enough, Itai? I can't see you, I hear you, but hopefully yes.
Yes, that makes sense.
Thanks. You can ask more.
So to fill the time, in case someone's writing out a question right now, I'll ask a question myself, although obviously a lot of that work was stuff that I did, so I can't ask specific questions about that stuff. But in a broader sense, when you think about interneuron dendritic properties in hippocampus, and when you look at network models that have come out, including all these different interneuron types from Perazzi's group, from Katsouridis, from Solstice, do you feel as though dendritic properties in interneurons have sufficiently been integrated into those models in terms of them making a difference to network output? No.
Well, I mean, that's why I sort of like I kind of really wanted to talk about strategy here. Oh, I don't know if I'm still sharing my slides. It doesn't really matter. But I think it really comes down to the strategy and what it is you're after. So I think, so my short answer is no. I mean, there's, as for example, there was that review which you probably know about from Schultz and Lassanzi, talking about deep and superficial layers and differences with pyramidal cells, and connectivity from different inhibitory cell types that sort of matters in superficial and deep layers. So we definitely don't have, those differences. And actually, there was another beautiful study from Klaus Kulander's group. I don't think I mentioned it here, but Mikulovic, that showed that the type 2 theta is in the ventral, is actually causal from the Orlan cell perspective. And so, and it's also, one of the certain frequencies, and then they cited this work about how does that come about? Well, it depends on the Orlan cell and what the biophysical properties are there. And then you have this distribution on the longitudinal axis of different inhibitory cell types. So I think there's, we haven't even in some sense begun to touch sort of the details associated with all of these details of the inhibitory cell types in the hippocampus and the cortex. And so that's why, I mean, I think these kind of studies that you and others have done is incredibly valuable, because, you could, first of all, to kind of get that data is really hard. And then once you have the data to build a model, not just build a model, think about the context, the rationale, the limit, why you're building it, and what you hope to get from it. And I think your work, I mean, basically has convinced me that this is a very fruitful way to go about trying to sort of figure out what's going on. And so even though we don't have everything in the dendrites in the ICE3, you just have sort of the KB 3.1 and all the rest of it, it sort of starts, it kind of gets there. And then one can start targeting and harnessing transcript, all the Allen-Bray study, but it's really a bridge. So that's why I really like that, our logo, developing bridges, because it's or even this patchwork quilt, which is, another quote from, say, Laria, but that's, to my mind, what it's about, right? I mean, it's strategies. We definitely haven't touched putting it in there, but we need to sort of figure out how and why and what is it we're after in terms of putting it in there. That's kind of how I see it. And so we need lots of smart, young, competent people to sort of carry on the work. That's how I see it. Yeah.
So there's another question from Itai. In terms of hippocampal functions, given how important hippocampus is for many things, we like this region too, smiley face, do the different functions involve particular circuits in different parts of the hippocampus? Does it influence your research design?
do the different yes, okay, so I'm looking at the question in the chat here too, because it's long and I'm just trying to make sure I understand. Do the different functions involve particular and different parts of it? So yes, I guess is the answer. So yeah, I mean, I kind of fell into the hippocampus, as John Lissman told me, this black hole. I didn't plan it. It was Liang's fault, who showed me this interesting data of hippocampus, and that involves sort of inhibitory coherence that sort of got me started there. So I guess for me, the way I've always thought about things over the years, coming from sort of the mathematical background, is I want the models to actually be helpful and tell us something. And so it's kind of always been that kind of thinking. And you know, so in terms of the function and what's going on, that's sort of, even, you know, we're not doing in vivo studies, you know, it's all in collaboration, right? The hippocampus is doing many things. I mean, it's essentially, as far as I'm concerned, the hub, and I'm not just joking. There's actually, you know, seems to be support for that idea that it's actually the hub. And so if we can kind of well understand the generation of these rhythms, whether it's theta, theta gamma, ripple, sharp wave, it sort of gives us a way to sort of bring about all of the functions. So I'm not sort of guided by, you know, I'm not coming at it from the behavioral functional perspective, but more from the mathematical model perspective, and knowing, you know, you got a 4-second on the system, you get chaos, right? And non-linear system. So you don't want to have things too detailed, but you need to put in some detail. So it's sort of like, where's that inflection point? So I am personally guided by the mathematical model and its limitations and what it could give us. I'm guided by that, but I mean, I'm not doing the model just because, you know, to have fun, although it is fun, but really, what can it tell us? And so I've tried my best to learn about the hippocampus over the years, just as I had to learn about somatic gastric ganglion, but really, to me, that's the only way. And so the theta focus has sort of came about because of the, you know, quite frankly, the Silva and Williams group that had this sort of intrinsic theta rhythm that they had in this whole hippocampus prep. So that was extremely exciting, as was Liang's work when I first got into that, because you had simultaneous access to a population activity and to a cellular activity. And because of that, you could actually constrain the puzzle, so to speak, of the mathematical model in a particular way. And so, you know, we had that sort of puzzle of the circuit with inhibitory cells, and then we started worrying about inhibitory cells and realized, oh my gosh, you know. So then once you get into the sort of those inhibitory cell models, like, okay, what is it that makes sense, or we don't know, that could contribute? So that's kind of how it is about. So I sort of, I'm not really asking your question, answering your question clearly, but to me, that's where I come from, so to speak, mathematically. So it's to make sure the mathematical model that we're developing is clear, the caveats and limitations, and helpful in some way. So I think actually the theta and the theta gamma and all the exciting work that is sort of coming out these days, you know, is a huge opportunity to really sort of link into sort of other functions, memory encoding, fair conditioning, all the rest of it. So yeah. So again, I don't know if I've really fully answered your question, but you can answer in the chat to say yes. I've really sort of given you my philosophy in some way, but I think that's why this sort of collaboration aspect is key, because, you know, whether it's Alex or Alexandra or Vlad or whoever's in the lab at a different time, they all have their sort of different perspectives, or they like, they don't like, you know, critical of, and that's really contributed to my own thinking of sort of what makes sense, right? I mean, I like people to, I shouldn't say argue with me, but sort of disagree, you know, as Alex has many times over the years, about sort of what makes sense, right? You know, because if we can't convince ourselves of, you know, the model and the helpful and the rational and the caveats, then yeah, you know, it's going to be helpful. So yeah, it's the key is that being helpful, and what is the best strategy and approach to use. Okay, I'll stop there. I'm kind of going on a bit too much, but yeah.
So I think that's all the questions there are none more. I'll pass it back over to Jess.
Thank you. Thank you, Alex.
Well, thank you so much for being here with us, Dr. Skinner. We had a really good attendee list, so we look forward to seeing you in the new year.
Thank you, and everybody stay safe and healthy, and thank you again for the invitation and opportunity to share my philosophy in some way. All right. Okay.
Thank you, everyone.
Okay. Thanks, everyone.